A very warm welcome to the participants of the Proteomics Advanced Winter School 2021. Presented by Proteomics Lab, Indian Institute of Technology, Bombay. Day 6, Quantitative Proteomics, Label Free. Lecture 3, Data Analysis for Quantitative Proteomics Experiments.
We are starting with this experiment, which we started in the morning today, about performing label-free quantification. And throughout the sessions, we are keep flashing you how the sample run is progressing. So I feel that you are all probably, despite being virtual, feeling that you are actually in the lab and you are doing the experiment, and how your experiment is progressing. And being a live run, you can see that not every run comes so great, and this run is did not start the chromatogram so smoothly, as one would expect, to go slowly and then reach to the higher intensities. But nevertheless, the run is still going on. And what we thought, we will, in addition to giving you this run and the data which we will provide you later on, we are going to show you some of the parameter part. So I'll just flash this little bit. If we get more time, we will also talk more about the parameter. But let me just give you this demo of some of the LC and MS parameters.
So like once you have set the method, once you have injected the peptide, like you have placed your peptide into the autosampler, the next point comes into the method development. So you have to develop a method for your run. So where we are using orbitrap fusion for our run and thermo nano LC. Now these are the important parameters which you need to know. First is the sample pickup and loading. Then is the sample loading, what is the maximum pressure that can be maintained. In case of gradient, here comes the main funda, like how much time of gradient you are using. Generally, my peptides are hydrophobic. Whatever peptides we are using is hydrophobic. I am using a C18 column. So basic logic is I am using two solvents. One is hydrophilic and the other is hydrophobic. That is A and hydrophilic, and my solvent is hydrophobic. When I am gradually increasing the percentage of my hydrophobic solvent, so whatever the hydrophilic peptides are there will be eluted first, followed by hydrophobic peptides. Okay. So this is the logic. We are generally increasing the hydrophobicity, and those which are more of the hydrophobic will be eluted at the latter stage. So here we are using an 120 minutes gradient, means 2 hours gradient, where my solvent B or my hydrophobic polar solvent is increased from 5% to 90%. So whatever the my peptides will be coming will be all eluted. So once the gradient has been set down, you will go to the pre-analytical and analytical column. These are basically the C18 columns. The maximum volumes which can be included for the pre-column is around 10 microliter, and that of the analytical column is around 8 microliter. And the maximum pressure which can adjust, like when the column pressure is existing 750, you will know that there is some problem. You have lot of unwanted materials in your solvent, so you have to in the peptide solution, so you have to again clean it. So max pressure is around 750. This is the 750 bar. This is the auto sampler position, where you can see like what samples are placed and what position. And this is the easy nano spray, like from here the nozzle will be sprayed. Okay. So now you move on to come to the main machine, like the Orbitrap mass spectrometer. In the Orbitrap mass spectrometers, you will have different parameters such as global parameters. In the global parameters, in the application mode, you can decide like whether you are analyzing peptide, small molecule, or an intact protein. We are generally using a peptide, so we have selected peptide. And the method duration, you have to sync it here. You have to make it 120 because we are using 120 2 hours gradient. Okay, this is method duration in minutes. You go to the scan parameters. In the scan parameters, actually these are the basic parameters which are generally used. In the scan parameters, there are various options starting from the MS, whether you want to do a full scan or selected ion monitoring, whether you want to do MS mode or DR, which is used for the targeted workflow, and various other sub parameters which can be used, such as the target mass used in the DR, means data independent acquisition. So in the MS Orbitrap mode, if you come to the MS Orbitrap mode, whatever the default charge state is set to 1, here need to define the postulates. Okay, here we are using a resolution, Orbitrap resolution of 60,000, but it is capable of going to 5 lakhs. You can see around 2.4 lakhs, here it is 5 lakhs of resolution. The scan range, what we are using is from 375 to 1700 nanometer. The scan range what you're using from the 375 to 170 nanometer. The RS range is 60. The edge target is 4.2 to the power of 5. Maximum injection time is 50 millisecond. We have set to 50 millisecond. You can optimize it according to your protocol. And finally, the polarity, we are observing the positive mode. But in case of metabolomics experiment, you can go for positive and negative both. The monoisotropic peak determination is kept to the peptide rather than the small molecules or the proteins, because we are analyzing mainly the peptides. The intensity, the filter intensity threshold, and the intensity threshold is kept in as the 5 point into e to the power 3. Charge state, we are observing multiple charge states, so it is around 2 to 6. Dynamic exclusion, like exclude after n times is 1, exclusion duration is 40, mass tolerance is we have set into ppm, not into m by z, and low and high is both as default parameters kept as 10. In MSMS mode, we have kept an isolation period of 2. We are using activation type as ACD. You can use CID and ETD depending upon your experiment. And we are using a collision energy mode as fixed. You can also step as stepped or assisted. Finally, we are using a detector type of ion trap. We are using a detector type of Orbitrap. You can use the ion trap also. And the Orbitrap resolution we have kept into 1500. Simultaneously, it is for the once the ion has been fragmented. So we are keeping as 15,000 in the detector type 2, which can be finally gone to, which you can include these parameters depending upon your molecule size. Okay, so once this is set, you can generate the method history. So once you are going for publication and all that, you can print this. You will know, like you can print this. Now you can know like whatever the parameters you have used in the document. So once this is set, we will incorporate this into the method. So you can see sample method have been lined up, and this is how the sample acquisition will begin. So this is from 0 to 120 minutes. Here we are increasing from 5% of B to 90% of the B, and you can see by the time it goes, the peptides are coming out, eluting from the column. You can monitor it on the real-time scale. You can see the peptides which are coming out, and you can also observe it remotely, like what is the quality of the peptide by clicking on leads. If one peak corresponds to more than 10% of more than 10 peaks of in MSMS2, like you can say that it is good fragmentation is happening. So you can see that one MS peak corresponds to more than 10 MSMS peak. Okay, so good kind of fragmentation is happening. And you can also see in the densities is way high, that is e to the power 9 as compared to the blank. When you compare to the blank, it is something, it is way high. So we generally, so for the instrument maintenance, we have generally set up BSA run, which we generally do, in-house BSA run for the daily routine checkup, whether the instrument is good or bad. So this is how chromatogram looks like. This.
All right. So you got some idea that there are many parameters to play with it here, and especially at the MS and MSMS level, you have multiple options to look into the collision energy, and that's where I think most of the time the major attention has to be paid regarding what is the collision energy you want to use. For instance, when you are using iTrack or TMT labels, you would like to play with that so that the proper collision happening for the reporter ions fragmentation at MSMS level. And so the number 35 what was given has to be played with, between 29 to 35 to see what works the best for your kind of labels and fragmentation. And also sometimes stepped works better, especially for the label based methods. So these things has to be played with it, and as we go along we will try to show you more software features. And of course, it will also depend on which instrument and which configuration you are using. That's why we are not spending too much time on this part, because this could also be very specific to whether you have a Q-TOF or a multi-TOF or you have Orbitrap. These parameters will change, right? So we are just giving you a flavor of it, but not spending a whole lot of time. But where we would like to spend some time is essentially data analysis part. Irrespective of which instrument type you're going to use, you are going to get some good MSMS data from different type of LC-MSMS configurations. How to now take that data forward and analyze them using public repositories as public software like MaxQuant. And also, as I mentioned, that you don't need to generate data all the time. You can go to the Proteome Exchange, download some of the data set of your interest, and try to probe a new hypothesis, new question from the available data set as well, right? And then you can still analyze using the MaxQuant and these kind of software. The major bottleneck in doing all this stuff is the space requirement on your system, as well as the speed and the power of the computing power. So for instance, we have shared you 6 files, which is almost 6 GB or so, which is going to be quite useful if you can actually download that in your system. And if your processor can process that, which is not going to take too much computing power, but at least you need to have the files with you. Nevertheless, we are going to show you the same file, what has been shared, 3 control and 3 treatment. And same we are going to walk you through with the MaxQuant steps now. We also understand that we will have to take a break at around 1:30 for the lunch. So we will start this process and then we'll resume back after post lunch at 2:30, where again we will finish off the MaxQuant and then continue with the proteome discovered session. All right. So let me now hand over this to one of my PhD scholar, Deepru Biswas, who is going to show you the data analysis part. We have already shared the Google Drive link. Please get it ready, and we are going to now show you MaxQuant.
Okay, thank you, sir. So I will first show the Google Drive link that has been shared with the participants. And I will request all of you that, as Professor Sanjeev has already mentioned, that you have to download these raw files. But in addition to these raw files, please download at least the demo MaxQuant parameter file and the Homo sapiens database. And there are a couple of study materials I have availed that you can also download those materials, which will be helpful during the next during the steps that we will show using the Max. So once I think last time we have already discussed regarding the downloading of the MaxQuant, and if I haven't you haven't seen that, so I will first request all of you that you go to the just write MaxQuant in the Google and right, just go and download this particular software. And meanwhile, I will try to give you some basics, which already Professor Sanjeeva has given during the MaxQuant. But how, the particular in MaxQuant, the things are pretty simple, as we have discussed. But how the things actually move when we are talking about complex samples and there are a number of multiple samples are there, and how these particular algorithms actually work. So it's a pretty simple workflow that I'm showing right now. So for example, you are having some protein samples and you have used an enzyme trypsin and you have digested those protein samples and you have a mixture of peptides. Then you have done some cleanup and finally what you are loading in the mass spec is the peptides, right? So when you are loading the samples in the peptide form. So the mass spec really don't know that which peptide belongs to which protein. So the most important part starts from here. The peptide, from the peptide, the mass spec will generate some MSMS spectra. And now these MSMS spectra need to be decoded, right? Because neither the mass spec nor the softwares that you will use will have any idea about which peptide belongs to which protein. So over here, the most important part of any kind of algorithm-based search or search engines comes is the database. So the database completely belongs which samples you are using. For example, if you are using some you are analyzing some human samples, then we need to download the human proteome database. And if we are using any other species, that make sure that we need to download the that particular data. So, and for downloading any database, I think all of you are aware now that we need to go to UniProt and we need to download that data. So once you are putting the database, this database act as a background, search base kind of thing. And once this particular spectra will convert into a peptide, and this particular peptide will search with each of the the sequence of the proteins, and it will get identified, right? And on the basis of that, then finally the proteins will get identified. So the proteins will get identified. And if there is any peptide which is coming from a different source for some reason, and the database and it is not present in the database, that particular peptide will not get identified. So to go a little deep into it, I will show you how this particular peptide matching is happening. So for example, you are having a peptide, and from that you are having a MSMS spectrum. So the particular, all these softwares, all these search engines, when you are putting the database, it's already making a theoretical spectrum. That means if you are having a protein, the amino acid sequence, the enzyme you are choosing is already giving the theoretical spectrum that which will act as a background database. And then this particular experimental spectrum matches with the theoretical spectrum. And finally, it gives a score based on this. That means a score based on how much the matches is happening. And on the basis of that, it will take the best match and it will also give the score. So this is how one peptide gets, identified using the theoretical spectrum or the experimental or the database that you provide, right? So this particular score generation and this particular, matching of peptides with the theoretical spectrum is something which all the search engines does, but the algorithms are pretty different. So over here only the difference in the algorithm, difference in the search mechanisms are actually coming, and why there are multiple search engines are there. And when we analyze same data, as Professor Sanjeeva told, sometimes we get a few of the proteins which are not matching with the database. When we are comparing the different search engine output data, right? So now coming to the database downloading. So we have shared the database already with you guys, but still, if you want to know that how the database is getting downloaded, we just go to UniProt, and then we search the human proteome over here, and we download the human proteome database as in a FASTA format, in a canonical form. So you can download this FASTA database. Make sure that we should check this particular database in after a couple of months, because after 3 to 4 months, the UniProt, for update its new database. Though there is not much difference in the proteome, but still it's better to use the new database, whatever the UniProt is uploading, which will help useful for our manuscript, because reviewers most of the time ask that which database we are using, right? And as I have already told that for the MaxQuant, you need to go to the MaxQuant site and download the software. It's a very pretty and a small software. And once you have downloaded the software, a MaxQuant folder will get generated. And over there is one icon will be there, which is the MaxQuant. So once you are clicking this particular icon, the application icon, so it will open the home page of the Max, right? So I think most of you has reached to this part, because it is a very simple part where we don't need to do anything much. So now I will try to show, I will just try to walk you through the simple steps of the MaxQuant, and then we'll try to discuss more about the parameter and in-depth way how MaxQuant works, right? So for this particular session, we are mostly talking about the level 3 quantification, and the raw files we have shared are not fractionated. So it's a single files, and how a simple analysis can be set, right? So for example, the first thing, so MaxQuant actually needs 3 important things. So first is the parameter file, the second is the database, and the third is the raw file. So first, when you are opening into a MaxQuant, if you're clicking into a file, you will find the load parameter and save parameter also. So as we have already shared the parameter file, just click on the parameter file, and I will request that all of you, if you guys can, follow us. So I will click into the MaxQuant. Yeah. So this is the MaxQuant. Yeah. So one more thing I just want to tell, that make sure whatever files we have shared, you are making one folder, and that should contain the database, the raw files, and the MaxQuant parameter file. So it should be there in the one folder. That will be easier for you. So once you are opening this particular file for MaxQuant software, make sure that you are clicking into the file and you are loading the parameter. So as you can see, the parameter, which is parameter file that we have shared with you, is available over here, and we are opening this particular file. So once this file, we have opened this particular file, the next thing is that what we will do is that we will load the raw file. So if you are already having some raw file showing over here, so you can select those raw file and click remove over here. So those are the residual files that MaxQuant shows, because we have shared our parameter file that we are using in our lab. So, and we will select all the raw files over here, and we will select insert. So as you can see, all the raw files are coming here. So these are the 3 tumor files, and these are the 3 control files. And the next thing what we will do over here is that, as you can see, there are multiple options there. So we are trying to make this particular session pretty easy and handy. So we are just showing for the level 3 quantification, and also for without any fractionation. So the next part we will do is that no fractionation will do. That means each samples belong to each samples have no fractions. But if you are having any fractions, then you have to click over here and you need to set the fraction. That means which samples belongs to which fraction. So for example, if you are having a sample with 6 fractions, you need to go to set fraction over here, and obviously first you need to select this. For example, these 3 are my fraction of one samples. Then you need to click select this, and then to set the fraction over, right? So this is the first thing. So what we have done till now, we have load the parameter and we have load the raw files. And the next thing is that we will go to the global parameter in the sequence. And what if there is any residual file is there, please remove those and then add the Homo sapiens database file that we have shared, or if you have downloaded that, you can add that. And this is the 3 thing what I showed: load parameter file, the raw files, and the database file. These are the 3 things that you need to do before starting the MaxQuant. So if these 3 things are all, these 3 steps have been followed, then you can just simply initiate the MaxQuant. So the reason I'm showing you these steps are that I just want to deliver this point, that how user-friendly the MaxQuant is, and the people who are just initiating proteomics, and if they are having any data which they have generated or they are downloading from Proteome Exchange or any other repository, it is very easy for them and very user friendly for them to initiate with the MaxQuant. But one thing, please make sure that the over here, as you can see, the number of processors. This is something which actually makes MaxQuant pretty powerful. That how many softwares how many processors you want to use. But make sure it is completely dependent on the system that you are using. Because if your system is having little less configuration, please check the number of processes you are having, and on the basis of that you can put the number of processes. If the number of processes is pretty high and your system is not compatible to those many processors, it might happen that the analysis get crashed, and it might be a problem for the PC too. Right. So for all for normal concerns, we will keep for 4 processors, because 4 processors is something which most of the PCs have. But still, I will suggest all of you guys to go ahead and check this particular thing, that how many processors you have. So once you have checked this particular thing of the number of processors, then what we can do, we can start this particular analysis. Right. Once we are starting this analysis, then it will take a couple of hours, depends on the configurations and the PC speed, that how much time it will take. and the analysis will be over. So this is the very simple procedure I show for level 3 quantification, and also for the samples which don't have any fraction, right? So now I can go back to my slides and I will try to walk through multiple. Yeah. So now. So I have already showed that, right? The folder should have one parameter file, the database file, and the raw file. And what the 4 steps for the level 3 quantification and the files without any fractions is that load the MaxQuant parameter file, load the raw files, click no fractions, load database, and click start. That's that much simple MaxQuant is for level 3 quantification. It's a 4-step procedure, and that's it. Your analysis will start, right? So these are some of the screenshots that I have taken, and I have already shown you guys, right, how these things will start, and I have talked about the processor, right? So now one very important and interesting thing which MaxQuant gives is that if you go to the visualization over here, and if you click here, it will give you something like this. So it might take some time for this visualization, but it will show something like this, which is a retention time versus M by Z graph of your each file, right? So it is showing that the retention time in the y-axis and M by Z in the x-axis. And trust me, this is a very good visualization figure that helps people who are doing optimization of their methods in using mass spectrometry, or they are trying to create a new method. It is very useful that how much that what should be the gradient time, and whether the elution is happening at the correct time or not, how much the M by Z is happening, how much of the proteins are coming in which M by Z length. So it's a very good visualization that MaxQuant gives, and as you can see, it is coming over here, and you can check multiple options are there. You can generate a report from here, and you can export this particular graphics, right? So once this particular analysis, I will just click in the start button, and I will request all of you to click, because I haven't talked about the parameters, which I will obviously talk about in this session. But I just want you guys to initiate the process so that the analysis will start. And once the analysis is, once you're clicking it, you can see the green bar over here in the right hand side. It is showing that the analysis in progress. And I will request all of you to click over here, the show all activities. So once you are clicking this, it will show all the activities that is happening one after another in the MaxQuant. And if you go back and try to see each activities, you will understand that how these activities are linked with the protein identification and peptide identification that is happening, right? So yeah, so as I have shown that how the run is happening, right? Yes, sir.
So you can mute yourself for a minute. To all the participants, I'm not sure how many people have actually reached to the downloading MaxQuant and going through the file, because it will be really useful now if you tell if you have any problem, and because we will walk through this step and show the demo. It will be a good idea if all of you are also able to do that in your system, right? So maybe during this break, please try to get the files available and download the MaxQuant. Reach where we are right now, then only I think it will be helpful for you to get more comfortable. So I think maybe we can take the next set of activities of parameters and things in more detail. You can just simply share the Google Drive link here, also the chat box, if some people are not able to see that, right?
Okay.
I just saw in one of the previous sessions somebody had asked question regarding the peptides for the non-nested peptide part. Essentially what you are looking at, that the peptide should be unique, and it should be sufficient in length, right? Around 9 amino acids or more, and it should be unique. But if you have peptides which are overlapping, so that means like this peptide comes from here to here, and second starts in the overlapping region. So then it is not going to be considered the 2 unique peptides. You want to have 2 peptides which are non-nested from different regions, and then they are unique. And that's going to increase the precision for the search in the databases and tell this is the right protein from the database search. So likewise, I think we should be now doing this activity together. And today this session, and of course the subsequent post-lunch sessions, are going to be more interactive with more assertion with the software, and we will see like how much we can do. If one software takes longer time, we can maybe show other software tomorrow, but we would also like to get people an idea that there are different software which can do the same thing but probably slightly different output. And that's why the reason we were giving you all of you same data file to be analyzed from one of the open access software, one of the commercial software. But if the time is not sufficient, I think we may restrict to only one software, and we will see like how best we could take this type of analysis forward, because what we want at the end, you take some small things from the course, but please be confident about doing those things, right? Like, can you now say that, okay, we're using the MaxQuant matrix science thing, I can analyze my simple file for angel digestion or identifying the proteins of interest, right? Do you have that level of comfort? And if you have that, then I think, then we have at least accomplished something. Likewise, if you take now three small data file of control versus, treatment, and this is a complex data analysis, what we are talking right now, where you have thousands of protein, and we are actually trying to do something which is in the real time, right? So we should try to do that. Of course, there will be many issues which I totally understand. There will be some software issues, sometime browser issues, sometime Mac issues, sometime system configuration problems. There will be problems. If you can overcome the problems by switching your system and borrowing somebody else's system or using something else, please do that. We will again resume back at 2:30 and continue this demonstration and talking more about parameters, but also about the output of the file and how to interpret those kind of data set. So these are all which we are going to cover in the post-lunch session. I hope you're all enjoying these activities, which we are trying to lay down for you. And also the run is getting over for the previous one, which I will discuss that how the results looks like in the subsequent session. Thank you, everyone. And I think you can take the lunch break now. We will again join you back at 2:30 p.m. Okay, that's all for the session. Thank you.